The overall goal of this procedure is to train mice with a forelimb-specific motor skill. This provides a good behavioral model to assess motor learning and memory in mice. The first step is to allow mice to become familiar with the training chamber and food pellets. Next, food pellets are put within reach of the shaping slit, and the preferred forelimb used for retrieval is determined. After the shaping period, the mice are trained with the single-pellet reaching task, and the number of correct reaches with the preferred paw is recorded. Ultimately, this protocol provides a behavioral paradigm to study brain changes at many levels during motor learning and memory formation.
This method can help answer key questions in the neuroscience field, such as how motor learning correlates with structural plasticity and molecular alterations in the mouse brain, and how various neurological diseases perturb fine motor skills.
For the best results, perform this task in a quiet location away from any noise or distractions. A custom-made clear plexiglass training chamber that contains 3 vertical slits is used for shaping and training. 1 slit is on the shaping edge, and 2 slits are on the opposite training edge. To hold the millet seeds in place during the shaping sessions, construct a tilted tray from 3 glass slides. A food platform is used during the training sessions. Divots on the left and right sides of the platform hold the millet seed in place. These divots correspond to the left and right slit in the mouse training chamber and are used for the training of dominant forelimbs. Other items useful to have on hand during the procedure are forceps, a scale, and a stopwatch.